So the purpose of this video is to go through and help you make corrections to your Year 10 biology mock exam from December. So we'll start with this question. Label the nucleus and the cell membrane on figure 1. A lot of students didn't even see this question because there weren't any dotted lines or boxes to tick or write on. So don't lose marks because you haven't read your paper thoroughly. So ideally get a ruler, and the nucleus is found in the centre of the cell here, the dark spot. And make sure you draw your line correctly showing that. And then the cell membrane will be the edge of the cell. Make sure it's very clearly pointing to the edge and not inside, otherwise that would be the cytoplasm. Then the cheek cells are a type of body cell. Body cells grow through cell division. What is the name of this type of cell division? The answer is mitosis. Differentiation is when cells turn into a specialised cell. So it is not going to be differentiation. Last question. What is the advantage of using an electron microscope to visualise ribosomes and mitochondria? Ribosomes and mitochondria are really small, so we want to use a microscope that is going to make the images a lot bigger so that we can see them more clearly, and that would be a higher magnification. The last part of this question is working out the magnification of a cell. So here we've got the cheek cell in figure 2 is magnified 250 times. I'd probably get in the habit of underlining information in the question, such as how many times it is magnified. And the width of the cell is shown by the line D to E. Calculate the width of the cheek cell in micrometers. Complete the following steps. So you need to get your ruler out and you need to measure the width of the cell using your ruler. And from D to E, it is 44 millimeters. A lot of students didn't measure this particularly carefully and got 45, but that will not get you the mark. So you write 44 millimeters. And then it gives you an equation to work out the real width of the cell. So you take the image size and you divide the image size, which was 44, by the magnification, which we have at the top of the question, which is 250. So get into the habit of showing your working. So 44 divided by 250, and the answer that will then give us will be 0.176 millimeters. The last part of the question then asks you to convert from millimeters to micrometers. And because there are 1,000 micrometers in 1 millimeter, we need to times our answer by 1,000. So 0.176 times 1,000 gives us the answer of 176 micrometers, and that's what you needed for the full marks. So you will always get a long answer question in your papers. This one was worth 6 marks, and it's done on a best fit basis, so it's not about getting a particular number of marks for each point you make. It's for the content that you give and how relevant your science is. And you're also going to be assessed on using good English, organising information clearly, and using specialist terms where appropriate. So white blood cells are part of the immune system. White blood cells help the body to defend itself against pathogens. Describe how pathogens cause infections and describe how the immune system defends the body against these pathogens. So this question has got 2 parts. The first part, describe how pathogens cause infections, and the second part, to describe how the immune system defends the body against these pathogens. So we'll start with the first part of the question to describe how pathogens cause infections in the first place. So I would start off by saying what examples of pathogens there are. So bacteria and viruses are pathogens. And how do they cause infections? They reproduce rapidly inside the body. And the way that they make us ill are one of two ways. So bacteria can produce toxins. So bacteria produce toxins. It's these poisons, these toxins that make us feel ill. And viruses actually live inside of our cells where they reproduce and then they burst out of the cell, killing the cell in the process. So they damage our own body cells. So bacteria produce toxins and viruses live inside our body cells, using our cells to reproduce, not multiply here, okay, because organisms don't multiply, they reproduce. They're producing more of their own species or their own organism. So bacteria produce toxins and viruses live inside our body cells, using our cells to reproduce. The new viral particles then burst out of the cell, destroying the cell. So that focuses on the first part of the question, describing how pathogens cause infections, producing toxins and destroying our cells by living inside of them. The second part of the question was describe how the immune system defends the body against these pathogens. Now, in the first part of the question, we're told that white blood cells are part of the immune system, and it's these that help the body to defend itself. So we know that we need to be talking about the specific immune response, not things like the skin being a barrier to infection or stomach acid killing microorganisms. It's about how the white blood cells defend our body. So there's various different ways. There are 3 ways in total that white blood cells help us to defend ourselves against pathogens. So the first one is that white blood cells can engulf the pathogen. So white blood cells engulf the pathogen and destroy the pathogen. That's called phagocytosis. The second way in which white blood cells can destroy pathogens is that they can produce antibodies. So white blood cells also produce antibodies, and these antibodies are specific to specific pathogens. So white blood cells also produce antibodies that are specific to the antigens found on the surface of pathogens. And what these antibodies do is they clump pathogens together, making it easier for other white blood cells to come and engulf them. So the antibodies clump pathogens together, making it easier for white blood cells to engulf them. The last way in which white blood cells help to defend us is that they can produce antitoxins. So white blood cells can produce antitoxins, and what these do is they counteract the effect of the toxins produced by bacteria. So white blood cells can produce antitoxins that counteract the effect of toxins produced by the bacteria. Then you might want to finish off by saying that this leads to immunity from that pathogen. This leads to immunity from that pathogen because once the white blood cell has seen a particular antigen on a pathogen, then it's able to remember that antigen and produce the correct antibody quickly, so you don't get ill. So this leads to immunity from that pathogen because once the white blood cell has seen a particular antigen on a pathogen, memory cells will recognize this antigen and make the correct antibodies rapidly. Okay, question number 3. So the heart is part of the circulatory system. Name one substance that is transported by the blood in the circulatory system. Again, reading the question is really important here because we had a lot of red blood cells are transported by the blood. Red blood cells are not a substance, they are cells. So you could have had any from glucose, or you could have had oxygen or carbon dioxide or urea or water or hormones. Any of those would have been acceptable. Moving on to the heart, the main type of tissue in the heart wall is going to be muscle tissue, because the whole point of the heart is to pump blood and muscles are required to do that. And we've got a picture of the human heart in figure 3. What you need to remember is a picture of the heart is going to be the other way around to how we see it. So everything on this side here is actually the right side of the heart, and everything on this side is the left side of the heart. So it gets swapped around. And just to talk you through what happens in the heart before we go through the various parts of it, blood arrives into the right atrium here, and this is through a vessel called the vena cava. So blood enters through into the right atrium where it flows down into the right ventricle. The heart contracts and the blood is squeezed up through the pulmonary artery all the way to the lungs, where that blood is going to pick up oxygen from the lungs and then is going to travel back from the lungs to the heart in the pulmonary vein. That oxygenated blood is now going to go into the left atrium. It will move down into the left ventricle when the atria contract, and then when the ventricle contracts, the blood is going to get squeezed up through the aorta to the rest of the body at high pressure. So when we're looking at which blood vessel, A, B or C, takes blood to the lungs, it's going to be B, because it's come from the body to the lungs to get oxygen, back to the heart now that it's got oxygen, and it's going to get pumped to the rest of the body through the aorta, which is C. Naming parts D and E then. So D is going to be the atrium. You could have written the left atrium. Atrium is one, atria is plural. You would have been allowed either. And then E is going to be the left ventricle. The next question, question number 4, is about the circulatory system and the fact that it contains arteries and veins. Now this one was actually out of 1 mark, and you had to describe how the structure of an artery is different from the structure of a vein. So describing is about saying what you see. The structure is what it looks like. And really importantly here, and I think this is where people did lose marks, is a lot of people did describe the structure of an artery, but they were not saying how it was different to the structure of a vein. So it has to have a comparison. So you could have had one of 2 things here. You could have said that an artery, so the artery does not have valves, whereas the veins do. And the other thing that you could have said was arteries have a thicker layer of muscle compared to the veins. This idea of thicker is really important because that gives you the comparison between the artery and the veins. If you just said that arteries have a thick wall or a thick layer of muscle, there is no comparison with a vein there, so you would not have been awarded the mark. Okay, question number 5 was generally well answered. This is about the human lung. So you are shown a diagram from the human lung, and it says that it has about 80 million alveoli. The diagram shows some alveoli in a human lung, and you had to give 3 features of the alveoli that allow large amounts of oxygen to enter the blood. Now, oxygen enters the blood through a process called diffusion. So you need to include anything that is going to speed up the rate of diffusion. A large surface area is going to speed up diffusion, and because there are lots of alveoli, there is a large surface area. So that would be for the first mark. So 3 features. First feature is lots of alveoli, give them a large surface area. The second mark is about saying that the diffusion distance is going to be thin, and that's because the alveoli have a wall that is 1 cell thick. Capillaries, which are around the alveoli, also have a wall that is 1 cell thick. So it's really important that you make it clear that it's the wall that's 1 cell thick and not the cell wall, because animal cells do not have cell walls. So the wall of the alveoli of the alveoli are very thin. They are only 1 cell thick. The last point that you could then make for the alveoli is this idea that they have many capillaries around them, and this gives them a very good blood supply. So there are many capillaries surrounding the alveoli. And that's the end of that question. Question number 6 showed you 3 diagrams. Diagrams A, B and C show cells from different parts of the human body, all drawn to the same scale. Which cell, A, B or C, appears to be best adapted to increase diffusion into or out of the cell? So we've already been talking about how diffusion can be increased, the idea of having a short diffusion path, the idea of having a large surface area, and the fact there are many capillaries surrounding the alveoli originally in the previous question means that you will have a high concentration gradient. So looking at these cells, one cell really stands out to the others, and that is cell B. And that's because B, the cell membrane has been folded many, many times into these structures here that look a little bit like villi. And the fact that it's been folded many times gives this cell a large surface area to maximise diffusion. So one reason for the choice being B is this cell has a larger surface area, and that will maximise the rate of diffusion. Question number 7. So the diagrams show the structures of a yeast cell and a bacterial cell. So we've got a yeast cell here next to the bacterial cell, and the yeast and bacterial cell have different shapes and sizes. Give one other way in which the structure of the bacterial cell is different from the structure of the yeast cell. Again, like the artery and veins question, is different from, we want some sort of comparison between the two cells. So you could have had one of several answers here. So I'll go through all the possible answers. So you could have written that the bacterial cell has no nucleus, whereas the yeast cell does. Or an alternative that you could have written was that in the bacterial cell, the DNA is free in the cytoplasm, whereas in the yeast cell, the DNA is found inside the nucleus. So that's just saying a similar thing to that first point. Or you could have noticed the fact that the yeast cell has got a vacuole, whereas the bacterial cell does not. So you could have written, or the bacterial cell does not have a vacuole, whereas the yeast cell does. Question 8 moves on to enzymes and digestion. And we've got a diagram that shows the human digestive system, and the question is all about where the enzymes are being produced. So digestive enzymes are made by different organs in the digestive system. Complete the table below, putting a tick or cross in the boxes. The first row has been done for you. So what's important here is it's asked you for ticks and crosses, so don't leave any parts of the table blank. So lipase is not produced in the salivary glands. So we would put a cross in this section. It is also not produced in the stomach, but it is produced in the pancreas and the small intestine. Protease, again, is not produced in the salivary glands, but it is produced in the stomach and the pancreas and the small intestine. And what's quite a good way to remember this is that the pancreas and the small intestine make all three types of enzyme. So they make them all, whereas the salivary gland only makes amylase, and that's where starch gets broken down in our mouth. And the stomach is only involved in the digestion of proteins, so it will be making protease only. Now this question, a few of you hadn't read it properly. The stomach also makes hydrochloric acid. How does the acid help digestion? So we got a lot of answers saying that the stomach acid kills the microorganisms in the food, which is absolutely right, it does, but that's about helping our body be protected from pathogens. It's not about helping digestion itself. So the reason why the stomach produces hydrochloric acid to help with digestion is that the protease in the stomach, it's called pepsin, so the protease pepsin in our stomach has an optimum pH of round about